Hello everyone, I'm Rajat Goyal and I'm presenting the journal ERP29 regulates epithelial sodium channel functional expression by promoting channel cleavage, which was published on June 18, 2014 in the American Journal of Physiology. A brief background of ENAC channels and ERP29. Epithelial sodium channels are called ENAC channels, which are located at the apical membrane of epithelia. They regulate the blood pressure, blood volume, sodium ion concentration, and the amount of liquid at the surface of airways. It is a heterodimer that has three subunits, alpha, beta, and gamma. ERP29 is a chaperone protein with a weight of 29 kilodaltons, where it gets its name, associated with the endoplasmic reticulum, which allows for the endoplasmic reticulum to function in folding proteins. It only has one cysteine amino acid residue instead of the typical two cysteines present at the motif of most ER proteins. The one cysteine in ERP29 is located at the 157th amino acid residue. Finally, ERP29 helps promote epithelial channel function. It also has a role in the formation of CFTR. Introduction. Decrease in function to epithelial sodium channels can be caused by siRNA mediated depression or overexpression of C157S ERP29. So what C157S is, C157S ERP29 is overexpressed mutated ERP29, which was mutated by taking the cysteine amino acid and replacing it with serine. Also, the Golgi apparatus is an organelle that packs and modifies proteins for secretion. And that'll be important later in the article. ENAC can bypass the Golgi apparatus and reach the apical membrane without Golgi apparatus processing, which has the capacity to cleave two of its subunits. If cleavage doesn't happen, the channels formed need to be activated to function properly at the cell surface. The purpose of the study is to understand how and to what degree does ERP29 regulate ENAC function, which is useful in the prevention of cystic fibrosis and hypertension. The hypothesis of this experiment was that ERP29 regulates ENAC biogenesis, trafficking, and as a result, functional expression. For the study, MDCK cells, or Maiden-Darby canine kidney cells, that had ENAC subunits tagged at the COOH terminal were used. Each subunit of the ENAC, alpha, beta, and gamma, had different tags and had inducible expression of ERP29. It was important that tags that did not alter the function of ENAC channel trafficking were used, and that's what the scientists did. And another part of the experiment was mutating ERP29, as was discussed earlier. Okay, so here are the results. So this is figure 1A and figure 1B. This figure shows the effect of ERP29 overexpression on ENAC in cells. As we can see from the control versus the dox in both the wild-type ERP29 and C157S ERP29, there's an increased expression with dox in ERP29. However, there's not really an increased expression in the other experimental samples of ENAC, alpha, beta, gamma, and GAPDH. On the right in figure 1A, we see how ERP29 has a positive effect in relative expression of ERP29 when treated with dox at a concentration of 50 nanograms per milliliter for about 8 hours. ERP29 wild-type ERP29 had an increased expression of 6 times, and C157S ERP29 had an increased expression by 6.6 times. Overexpression of wild-type ERP29 increased the relative expression in the beta domain of ENAC channel by around 2.5 times, which we can see from in the third sample over on the x-axis. and the alpha ENAC and the gamma ENAC full-length expression seemed to be unaffected. The low-band gamma ENAC had a positive effect as well by approximately 1.6 times. However, in the overexpression of C157S ERP29, it reduced expression by 40% in the gamma ENAC low band and left expression in the other domains pretty similar. So a negative effect in the last column right there. And figure 1b showed the same results as figure 1a, but the experiment was performed in a different way with the use of a plasmid which encoded for wild type ERP29 and C157S. And it showed the conclusion again. And in figure 1b, we see the conclusion docs increased the expression, but the increase in expression was only about 3.3 times in the wild type ERP29 and around 3 times in ERP29 C157S. So siRNA negatively affected ERP29 expression by 60% decrease, 40% expression relative to control, which correlated with a 60% decrease in ERP29 expression in beta ENAC. Expression of alpha and gamma subunits did not appear to be unaffected here. So right here we have our unaffected gamma and alpha units, but ERP29 with siRNA decreased by 60% relative expression right here at 0.4, and so did the beta ENAC right here, black. So conclusions. Doxycycline, which is also called DOX, increased the expression of ERP29 expressing cells in both ERP29 wild type and C157S. ERP29 when compared to the control for all the experimental results. siRNA negatively affected ERP29 expression in ENAC. So these the next next four are more important conclusions. Cysteine of ERP29 had some role in expression and regulation of ENAC. Increasing ERP29 expression resulted in an increased expression of ERP29 in ENAC. ERP29 expression correlates to expression of ERP29 in ENAC subunits, and ERP29 regulates whole cell ENAC expression in MDCK epithelial cells. So regulation of ENAC would allow for the prevention of cystic fibrosis and hypertension. ERP29 regulates and interacts with epithelial sodium channels in the cells MDCK and CFBE41 and in xenopus oocytes, and these are the three types of three types of cells where this experiment was performed by these researchers. ENAC functionally depends on two things. So the channel open probability, so how likely is it for the channel to be open, which is indicated by PO, and the total number of channels present at the cell surface, which is indicated by N. And it was found that ERP29 increases probability of functional ENAC at apical surface of channel and not the number of channels, N. So cysteine in ERP29 plays a role in ERP29 function. Overexpressing C157S, ERP29 decreases ENAC functional expression. And SNI, and this was a similar result found in siRNA-mediated depression of ERP29. ERP29 directs ENAC towards the Golgi, where it undergoes cleavage during its biogenesis and trafficking to the apical membrane. And ERP29 promotes delivery of ENAC that go through Golgi processing while reducing those that don't. And you guys are, what role does ERP29 play in the prevention of cystic fibrosis? How does it do this? Why is it important for ENAC to be processed by the Golgi apparatus before it reaches the apical surface of epithelia? How does ERP23? And here's my source.